So I gotta do this.
Good morning, everyone. I hope you're all here, bright-eyed, bushy-tailed, and ready for a great day. I'm going to have a couple of housekeeping announcements that I need to make. And I accidentally closed that note on my phone, so give me 1 second. Everything on your app or your program listed as Aurora 4 will actually be Aurora 3. So make sure that if you're looking for that, you're looking for 3, not 4. And the store is going to be open till noon. And I think that is the end of my little housekeeping announcements. Just keep in mind, I know we've got a full schedule again today. And again, the break stuff will be out for y'all. Come and go as you need to. And I think I will get us started. Dr. Benke is going to be talking this morning. And he had a nice little creative name for his talk. And so I have closed that out on my phone now. And it is, CDD disorder. I don't have to read it to y'all. Y'all can read. He's great. I'm going to go ahead and introduce the hour so it moves a little quicker. So right after Tim, you'll be hearing from the Lulu Foundation, and Majid Jafar will be talking about that. So I hope you all have a wonderful day. And they, again, were our travel grant sponsor, the reason we have so many representatives from all the different countries. They're wonderful people. I hope you've met them. And y'all enjoy.
Thank you, Karen. So I'm going to apologize to you because it's 8 o'clock on a Saturday morning, and I'm requesting total engagement of the brain. So I don't have anything relevant to disclose for this morning's talk. So I'm going to give you a quick overview of how the brain works. Then I'm going to do a PubMed search and go over some, how I've been approaching the literature. And I'm not going to do all of the CDKL5 literature. We can't do that. But basically, what are the highlights from about 2016 to the present, which is about the last time we had a meeting, here in the States. So what are the types of research that I've reviewed? So there's basic science literature. So these are studies to investigate the basic biology of what CDKL5 does. Then there's translational science. So these are studies in typically rodent or cell-based models, or even zebrafish or worms, about how CDKL5 deficiency might affect how things work, and in some cases, to look at how potential therapies might or might not be effective. And these are where we get a lot of our excitement that's going to bring us forward to clinical trials. Then I'm going to talk about just a few studies because there have been some good ones, but I think some of them you've already heard about at this meeting, so just the ones that you may not have, about studies in humans with CDKL5 deficiency. For each one, I'm going to talk about their approach, the results, their general conclusions, and then torture you with some of my musings about where this, we need to think about this from a critical scientific perspective. And so I think that's important because we don't want to say, oh, this is going to be great, but we need to, make sure that it's really going to move things forward with how we take care of children. When I go to work every morning, I say, please, God, please don't let me hurt anybody today. And I think this is the basis for that we have a sound foundation to move forward with how we take care of our children. So I don't think there's I pared this down a lot. It's just a couple of papers because we have less than 30 minutes to talk about this. So briefly, cyclin-dependent kinases or CDK-like kinases. So you have to ask yourself, why do they call them like kinases? Are they teenagers? Is this some sort of valley speak? Why did they put like in there? So cyclin-dependent kinases, they regulate mitosis. That's how cells divide. They regulate the growth functions of this other thing called primary cilia, which is something how cells move. And the cyclin-dependent kinases bind and are regulated by binding of these things called cyclins. Cyclins are what help cells cycle through the different cycles of their life as they make new cells. But cyclin-dependent kinases, they have a cyclin binding domain, but they don't bind cyclins, so that's why they're like. And so we really don't know what cyclins do. So let's talk about, let's see if I can get a pointer to work here, but basically just look at the top half. But on neurons, there are these little things called dendritic shafts, and they have these little protrusions called dendritic spines. And dendritic spines and synapses are the chemical bridge between electrical communication from one neuron to the next. So it's a chemical bridge in communication. And so you have different types of synapses. You have excitatory synapses and you have inhibitory synapses. Oh, that's going crazy now. I was just trying to figure out, is there a pointer there? No, I can't get a pointer there. So basically at the bottom half of the screen is one of these spines on the bottom and one of the axons on the top. So electrical information will come into that axon. It will cause those little circular things, which are vesicles that contain neurotransmitter, to be released. That neurotransmitter will kind of go across that little space between the two, and it's going to bind to receptors, which are the little red or orange cones that are there on the other side. Now, these are excitatory neurotransmitter receptors, and so you have different flavors. You have the NMDA type of receptors and the AMPA type of receptors. And I'm telling you this because there are studies that are looking at changes in both flavors of those receptors. And what's also important is it's not just whether or not you're an NMDA receptor or an AMPA receptor, it's the subtype of that receptor which is also important about how the synapses function. So that's why I'm telling you this. But the other thing is if you see where those receptors are, they've put this nice little yellow haze around those receptors, so something's holding them in place. That something is called the postsynaptic density. And there's a lot of different proteins that we call scaffold proteins that are holding them all in place. And, in other words, if you don't hold one of these receptors in place, they're going to go away. So you've got to hold them there. And they're basically one of the other things that's also sitting in these synapses, which is the little purple plus sign down in there, the kinases. CDKL5 is one of these kinases that's sitting at synapses. So the other type of synapse are inhibitory synapses, which are in the upper left-hand corner there. These actually release a different type of neurotransmitter, which is called GABA, typically, or glycine. It does the same sort of thing, but it doesn't do it onto a spine. It does it onto the dendritic shaft. And it binds to a different type of receptor. It binds to GABA receptors. And basically, in the lower panel down there, I'm trying to diagram what the difference is between an excitatory synapse and an inhibitory synapse. Excitatory synapses allow the flux primarily of positive ions, which depolarize a neuron, whereas inhibitory synapses allow the flux of negatively charged ions, such as chloride, which hyperpolarize a neuron. And this sort of balance between depolarization and hyperpolarization is a balance. And so in the upper corner, I've got a balance between excitation and inhibition. And this is something that's kind of in vogue in terms of how we talk about synapses, is this balance between excitation and inhibition, the EI balance. Well, if you have too much excitation and too little inhibition, you can have a seizure. But if that balance goes the other way, you could have intellectual disability. And it depends on which circuit this is happening as to whether or not you're going to get one or the other. So it's not the same throughout the brain. It depends on which circuit we're talking about. So that's that initial gets you in there. Let's think about how all of this works. So this is just kind of an update up to 2016. But basically, we know that CDKL5 is localized to these dendritic spines of excitatory glutamatergic synapses, as well as the nucleus, which is the instruction center of neurons. CDKL5 is then thought to be a kinase, that is, it puts a phosphorylation group and regulates MECP2, which is the regulator associated with Rett syndrome. It stabilizes the formation of synapses by interacting with one of the main organizers of synapses, postsynaptic density protein 95, and then there's a long list that's ever growing here. We wonder if these could be the cyclin-like equivalents that it binds to. The kinase so CDKL5 is phosphorylating other proteins. There's an emerging list of who it's phosphorylating. Netrin G ligand 1, amphiphysin are some of those that are on the list. And when you take away CDKL5, there's been loss of excitatory synapses in some model systems. And one of the models that's been presented is this model here at the bottom, that CDKL5, it's binding to the little yellow thing, which is PSD95, but basically when it's not there, PSD95 can't do what it's supposed to do at the synapse, and then there's dysregulation of AMPA and NMDA receptors. So let's jump into the literature. So basically, there was a paper that came out from Edinburgh, led by Ralph Hector, and he investigated CDKL5 variants. It was entitled Improving Our Understanding of a Rare Neurologic Disorder. This was in Neurology Genetics. And basically, the punchline from this paper is that missense mutations, and you had the talk on Genetics 101 yesterday, these are the ones that don't prevent you from making the whole protein, but they're little misspellings. But it's those misspellings that are in the kinase domain are probably the ones that are important. And basically, Ralph did this by mapping out all of the different missense mutations to try to understand how this could be. So then there was another paper that came out in Cell Reports. This was titled CDKL5 Family Kinases Have Evolved Distinct Structural Features and Ciliary Function. So this was a basic science paper. And what they did is that they provided us with the crystal structure. So what does the three-dimensional picture of CDKL5 look like compared to other CDKLs? And they did the crystal structure and compared it to the other CDKLs. What they did, though, is they really just looked at the kinase domain. They didn't look at the whole protein, which is very important. But they were just interested in the kinase domain to see what that looked like in a three-dimensional picture. They looked at worms. It's a very useful system called C. elegans and cell lines. And they investigated cilia. So these are these cellular protrusions from cells. Neurons do have what they call a primary cilia, but we don't know what it does. But we do wonder if the same machinery that's in that primary cilia is also something that causes spines to form. We don't know that yet. But anyway, these cilia, they're cellular protrusions. They're the motors that move stuff around inside cells. They investigated CDKL1. It's not the closest match to CDKL5, but they put CDKL5 mutations into CDKL1 and see what it did to the worms, basically. And at the end of the day, it's unclear how ciliary functions translate to the vertebrate nervous system, and it's unclear how well the CDKL1, which is really a short CDKL5, really models CDKL5. But basically, normally you get a regular-sized cilia in worms with normal CDKL1, but if you overexpress CDKL1, you get these short cilia. But if you put in patient-related mutations in the kinase domain, you get these long cilia. Okay, this got cut off at the top, but this was an exciting paper that came out of from the Japanese group by Dr. Tanaka's lab, and their paper was CDKL5 controls the postsynaptic localization of gluin 2B containing NMDA receptors in the hippocampus and regulates seizure susceptibility. So that's a mouthful, but it goes back to that notion of NMDA receptors and a certain flavor of NMDA receptors that's involved. So this was translational science, and the Japanese group, basically they generated a new, different mouse model of CDKL5 knockout that's different from the one that's been used so far, and they looked at male mice. Again, they didn't find spontaneous seizures in these male mice, so of course very different from what we know in the clinic. There were no spontaneous seizures, but they wondered, well, what if we try to induce seizures with a chemical proconvulsant? Is there a lower threshold? And basically, when they gave something called kainate, which tickles AMPA-type glutamate receptors, the seizure susceptibility was the same. But if they gave NMDA, which is the chemical that tickles NMDA receptors, they found that there was increased seizure susceptibility to NMDA. They thought, well, that's interesting. Maybe there's something weird with NMDA receptors. So they then took out hippocampal slices. You can make these slices live in culture for a long time. and they studied how the synapses work in these slices. And they studied something called long-term potentiation, which is a cellular model of how synapses learn in a dish. We think it's equivalent to learning and memory. And interestingly, they found increased long-term potentiation, which normally you would associate with loss of learning and memory. They found that there was an increased contribution of NMDA receptors to synaptic currents, and they were able to pharmacologically attribute this to the gluin 2B flavor of NMDA receptors. They found that this was due to an increased surface expression of these gluin 2B receptors. The question is does it really help us explain, the EI imbalance that we know exists in CDKL5 disorder? They looked at it in hippocampus, but it really is, the question is this present in other circuits? And again, there's this question of it doesn't really fit. There's increased LTP, but the same group also published a paper that they have reduced learning and memory with this mouse. So it's not really fitting together in the way that it should fit in my head. But it also needs to be placed in the context of deficits more broadly in this mouse model. And you can't read down at the bottom there, but basically we need to look, if this is the case, in the other mouse models of CDKL5 deficiency to see if they also see this enhancement of gluin 2B. The next paper, and I wanted to keep this paper in here because there was a question that came up with our discussion yesterday. And so this came from the European Journal of Neuroscience from Elisabetta Ciani's group. Treatment with GSK3-beta inhibitor tidaglutide improves hippocampal-dependent and memory performance in juvenile but not adult CDKL5 knockout mice. So this, of course, is a translational study. They investigated CDKL5 knockout male mice. As before, what this group has found is that GSK3-beta, this is another type of kinase, had increased activity in the knockout mice compared to the wild types. What they then did is they treated for 20 days with this GSK3-beta inhibitor. It normalized GSK3-beta activity in the hippocampus in both 3-week-old mice and in 13-week-old mice. But only in the 3-week-old mice was it found to improve learning, memory, dendritic structure, synaptic contacts. So it suggests that early treatment with this paradigm is important. Again, this is the sort of thing we need to see if this happens in other people's hands, if they see the same thing with these types of drugs. What I think we need is we need more clarity on the signaling pathways that lead to GSK3-beta activation, because we just kind of have CDKL5 in our diagram up here, but all of the arrows that lead to how it interacts with GSK3-beta, we don't know how that necessarily works yet. And again, we need to have better clarity as to what this is doing in other circuits other than the hippocampus, because clearly other circuits are going to be impacted because you're giving this drug to the whole animal. Okay, now this is a good one. So this also came out of Dr. Ciani's group. So this was in human molecular genetics. CDKL5 protein substitution therapy rescues neurological phenotypes of a mouse model of CDKL5 disorder. So this is translational science. They investigated CDKL5 knockout male mice. These were 16-week-old adult mice. What they did is they took that big, huge CDKL5 protein and they put an extra little bit of protein on the end called a TAT cap. And what is TAT? TAT is actually a protein that comes from the HIV virus that can help proteins get shuttled across membranes. So it's not a normal thing for it to be there. It makes a protein cell permeable. Now, what's really remarkable about this study, which for some of us who used TAT-capped proteins, is they put the TAT on this huge protein. In my lab, when we do studies with TAT-capped proteins, we are only able to get proteins this big to go across cell membranes. And so it's remarkable that they were able to get this result for such a huge protein. And it crossed the blood-brain barrier, which is just, it's remarkable for those of us reading the paper. So, but they found that this protein, it improved learning and memory, improved abdominal dendrites, breathing, and clasping phenotype in the mice. In order to get this work, of course, they put it in these little things called mini pumps that are implanted underneath the skin, and so it releases the protein over, weeks. And so it required, this sort of continuous infusion. But of course, this sort of finding, it needs to be validated in other labs. Can other people get the same result? The reason why, we say this is just, this is the way science is. It has to be validated and done in other labs because it's just, everybody does things a little bit differently. Everybody's mice are treated a little bit differently. How you do the behaviors, it's a little bit different. So you got to be careful about, you can't just say, this is it. So it needs to be validated. The other issue is that apparently it's really hard to make the CDKL5 protein in large enough amounts to do these sorts of studies. So that's a big deal. You need to get around that technical hurdle in order to try to redo this as well. I'm conscious of your time, Majid. I'm not going to take your time. So another paper that came out is the antidepressant tianepine reverts synaptic AMPA receptor deficits caused by deficiency of CDKL5. This is a paper that came out of Charlotte Kielstrup-Nielsen's lab, was in human molecular genetics. It's translational science. So what they did is they looked at rat hippocampal cultures. So basically, if you take out the brain of a rat, in this case they took specifically the hippocampus, if you grind it up and put it out on a culture dish, it actually grows and reforms its connections. And so they knocked down CDKL5 right after they did this. And then they treated the cultures after they'd been in vitro, living on the dish, from day 11 to day 18. They found that knockdown had reduced surface expression of two flavors of the AMPA-type glutamate receptor, GLUA1 and GLUA2. But this ratio of how much GLUA1 or GLUA2 was altered. And that ratio is important because it regulates how the cells signal. That actual change in the ratio can potentially make the neurons hyperexcitable. Anyway, this treatment normalized that ratio, which is exciting. But basically what we need to do is, there's some limitations here. So this really is modeling early synaptic development in an unknown class of hippocampal neurons. When you plate hippocampal neurons out on a dish, you don't know if they're excitatory neurons or inhibitory neurons, and this is a knockdown versus a knockout. So these are things that need to be validated in other rodent non-culture models to see this is really the case. And of course, the same issue that I bring up was we need context in terms of circuit dysfunction and to see if this is the case not just in hippocampal circuits, but say cortical circuits. I think I'm getting close to the last paper too, but this is also another question that came up about what about X inactivation studies. So this is a mixed modality approach towards XI reactivation for Rett syndrome and other X-linked disorders. So this was in Proceedings in the National Academy of Science from Dr. Lee's lab. So basically, I'll call this semi-translational science because it's proof of principle before you kind of move on. So XIST is a protein that's the master regulator of X chromosome inactivation. And so first they did a combination approach. They used this thing called an antisense oligonucleotide to reduce the levels of XIST, plus they added a chemical inhibitor of DNA methylation. So in vitro, so cells growing in culture, when they did this, they found that there was a little bit of an increase, 2 to 5% in MECP2 mRNA. So what we were looking for is, a 50 or 75% increase. In vivo, they tried this as well. Again, this was in whole animals, but it was a bit of a trick because they did a knockdown of XIST, plus they gave the animals an inhibitor of DNA methylation. They found a 5% increase in MECP2 mRNA. So the limitations of this is it was an artificial knockdown of XIST. But, if you were to apply intrathecal antisense oligonucleotides, you could do similar perhaps. And they haven't yet applied this to a disease model to see if this really works. And the big question is would a 5% increase in MECP2 or CDKL5 really be enough to ameliorate symptoms? So while we're still hopeful that we can understand X inactivation a little bit more, it sounds like there's more to go with the science before this becomes a viable thing to pursue. This was in my title, but I'm going to skip it for the sake of time. But basically, zebrafish are being studied. Zebrafish are exciting because you can make a lot of them real quick, so they could potentially be a model that you can use for high-throughput screening. This one, let's talk about. So this is from Joe Zhao's lab at UPenn. Loss of CDKL5 in glutamatergic neurons disrupts hippocampal microcircuitry and leads to memory impairment in mice. So this was translational science. This was mice with knockout of CDKL5. But the genetic trick they did is it's only in forebrain excitatory neurons. I left out the excitatory word. But basically, they wanted to do this to address the question of whether CDKL5 loss causes circuit dysfunction in a cell-autonomous fashion. So if you have CDKL5 loss in one neuron, does that influence the other neurons that talk to it or it talks to? Or is it really just dependent upon that neuron involved? So that was the question that they wanted to address. So they found that isolated hippocampal-dependent learning and memory was altered, but other tests of behavior were not altered. There were no seizures in this mouse. But basically, there is a cell-autonomous function of CDKL5. So loss of CDKL5 increased. So look at the green triangle on the left there. That's a neuron that's lost CDKL5. And there's this red neuron that's talking to it. That's an inhibitory interneuron. So the green cell was the one that lost CDKL5, not the red one. But when you, in the next panel over to the right there, so when you lose CDKL5 just in the green neuron, suddenly the wild type red neuron is making more inhibitory synapses onto that. So basically, one interpretation could be that cell-autonomous loss of CDKL5 is causing more inhibitory synapses to be formed onto that cell. But at the same time that they found this, there was also more excitatory synapses that were formed onto the cell. So it seems like there's a mix of cell-autonomous and circuit-related dysfunction that's leading to an alteration in the number of synapses. So the big question is, you know, what's really cell-autonomous and what really isn't? So we have some information, but we're still asking a lot of questions there. And the big question is does this explain EI imbalance? I'm not sure that it does yet. The other question is this was done in hippocampus, but these same animals can be studied in the cortex, so I think it's important to look at the cortical circuits as well. Okay, this is the last one, I promise. So this is a paper that if you use PubMed, it doesn't come up. It's only in the fine print. So the title of the paper was Identification of an Elaborate Complex Mediating Postsynaptic Inhibition. It's from Scott Soderling's lab at Duke. And this addresses the basic science of CDKL5 function. And they were investigating young adult mouse brain. So they did a trick where they put this little tag on PSD95, which is that thing that holds excitatory synapses together, or they put a little tag on Geoffrin, which is what it holds inhibitory synapses together. And this little tag is going to label any other protein that is within 40 nanometers, that's really small, close to those other proteins. So basically you can come up with who's talking and touching whom in the synapses. We call this an interactome. Basically, it had the power to identify proteins common to both. And this is kind of a holy grail because we know that 25% of spines also have an inhibitory synapse on their neck. And so if we know the proteins that kind of mediate this crosstalk, that would be really important in neuroscience. So they gave us this nice little picture of the interactome. And you can't see anything in there, but basically the size of the circles is how strong you're talking to somebody else. But when you look really closely, there's our favorite protein that's part of the inhibitory synapse interactome. And they also did this with PSD95, the excitatory synapse interactome. And, of course, there again is our favorite protein right up there at the top that's talking to both. So CDKL5 is a common protein to both inhibitory and excitatory synapses. And I think that the next step is to use this technique to determine specifically the CDKL5 interactome and to look at it in other circuits like cortex, striatum, etc. So I think this is really exciting. So that's all I have for you. I've given you a fire hose of just a couple of papers. I hope I didn't explode your minds, and I'm happy to take other questions, but we started a little bit late, but I tried to catch up. Catch questions at the end. Great. Thanks very much.
Hey. Good morning. I'm going to put my watch here. It's great to be here. We've got half an hour, and my purpose this morning is just to give you a little bit of an introduction to the Lulu Foundation, to the work we've been doing, and I'll try and finish in 20 minutes so that we can take some questions as well. First of all, I wanted to introduce the team. To my right immediately is Lean, my life partner in everything, including this endeavor, who co-founded the Lulu Foundation with me. To her right, Dan Lavery, who's our Chief Scientific Officer. We hired Dan from the Cure Huntington Disease Initiative, which is a leading organization tackling that rare neurological disorder, and Anna Mignorance that you heard from yesterday. And Dan is US-based and Anna is European-based as our Chief Development Officer. And yeah, sorry, we're Dubai-based, but the foundation is actually UK registered, so we kind of hopefully have our bases covered, almost. So start with the patient, always starts and ends with the patient. This is our eldest daughter, Alia. This is why we're here. And our journey is the same as yours. As Rick beautifully said yesterday, it's only this one thing that unites us, but it is everything. Her first seizure started when she was 19 days old. She's our first child, and we went from the highest high to the lowest low experience that you all know well, through the saga of diagnosis, trying different drugs, multiple anti-epileptics, the honeymoon period, spasms, steroids, more anti-epileptics, and then, of course, the diagnosis, and to be told she's missing one letter from one copy of one gene, which is like missing one letter from the full set of the encyclopedia, 30-volume encyclopedia. But because of that, she will probably never walk or talk and have seizures every day for the rest of her life. And we went through the shock and the sadness, but reaching out to IFCR and Catherine and David Frame, Catherine, who we had like a 3-hour onboarding on YouTube, which was really helpful. We suddenly found this link with people from the other side of the world that we had more in common with suddenly than our own family and close friends. Alia is now 4 years old and still can't walk or talk and has daily seizures, and yet we are happiest when we are in her company. I know something that all of you can relate to. So this you know, largely CDKL5 deficiency disorder. This is kind of where we're at. The latest estimates of the incident rate are 1 in 40 to 50,000, which may sound rare, but as rare diseases go, it's actually not that rare, and it's it's actually at a sweet spot for industry interest. And it's becoming clear that this is probably the second most common genetic form of epilepsy after Dravet syndrome. It's about half as common as Dravet syndrome. So we know of about close to 2,000 medical cases, but the true number is many, many times that. And the frequency of diagnosis shoots up because the genetic test is more available, and so the average age of diagnosis is now well below 1 year. So it's much more common to have babies diagnosed than just a few years ago. And clearly we need better treatments and cures, which led us to form the Lulu Foundation. Our first engagement before we'd formed it was actually through IFCR, and we made a donation to IFCR to advance some of the research. And then we chose to establish the Lulu Foundation, not as a patient organization, because already they existed in all the main countries, but to focus on translational research towards therapeutics. And really the overall mission, if I were to summarize it, is to de-risk this disease, to make it attractive for industry, for companies, and to pursue multiple approaches, and to run with parallel objectives. So funding academic research is the main thing that we do, certainly, and we have we funded some long-term grants, some of them in partnership with IFCR, and we have a pilot grant program in partnership with the our strategic partner, which is the Orphan Disease Center at the University of Pennsylvania, which is where Dan is based. So he's our CSO, but he's also director of that research program it's called the Program of Excellence for CDKL5 Deficiency. Engaging and enabling industry is critical, and I just wanted to quickly go through and introduce some of the sponsors on the list up here. Marinus, you heard from in detail yesterday. It's a small company with a big heart, and a huge thank you to them, not only for developing this drug and choosing to focus on our disorder and our kids. Alia is actually on the drug now as part of the phase 2. But also breaking ground for us. By doing the first phase 3, they introduced this disease, this disorder, to the FDA and the EMA. I mean, you may think that they know everything, but they don't. There's 7,000 rare diseases, right? So actually it's on us and the companies we work with to educate the regulator. So thanks to them, the regulator very well knows now this disorder, that it's a high unmet clinical need, and they're really willing to work with us to try and accelerate new therapies, new therapeutics. And I can also announce that the Marigold website is now live. It came live about 10 minutes after Lorianne mentioned it, but that's an age in the digital world, so it is now live. Takeda, we heard yesterday from Mahindra. That's a big company with a big heart. And actually it's just become twice the size, hopefully, by acquiring Shire, the biggest rare disease company in the world. And that'll put them in the top 5 of global pharma giants. But just amazing people, so multinational, so patient-focused, so externally focused, and they partner with patient associations, with other companies, rare disease companies. The chief scientific officer, Andy Plum, is a wonderful guy whose neighbor and good friend actually is a CDKL5 dad, or former neighbor. So he spent 10 years living next to a girl with this disorder, and he actually traveled around Takeda offices, I heard recently, showing a video of a child affected with the disorder and pointing out to their staff about what they're doing for us, which is amazing. Greenwich Biosciences, or GW, as they're known in Europe. Again, a great company you heard from yesterday, the company behind Epidiolex. Amicus Therapeutics, a company led by a rare disease father, John Crowley. You've probably seen the movie Extraordinary Measures. They've acquired the program you just heard Tim talk about the enzyme replacement therapy, and that's thanks to a startup that was established by Mike Jasulavec, who's another CDKL5 dad. He was here yesterday. He headed back last night because it's his daughter's birthday today. But we are very grateful to the entrepreneurship that he did, which led to Amicus now having CDKL5 protein enzyme replacement therapy as their leading preclinical program. Ovid, you heard from again yesterday, great company, great CEO who made his name in big pharma but chose to focus on this rare disease company in the neurospace, neurodevelopmental disorders, and also happens to have a close friend who last year had a grandson born to his daughter who was affected with CDKL5 deficiency. So he's personally affected as well. PTC Therapeutics, they were actually the first to do a small exploratory trial for nonsense mutations. So huge thanks to them for taking that step. Novartis, another giant in the pharma world, they are actually now interested through an academic program we funded at Harvard and Mike Greenberg's lab, and they've also acquired Avexis, the company you heard about yesterday from Tim, which is the company that led the SMA gene therapy. Roche, head of rare disease at Roche, is actually Omar Khawaja, whose photo you briefly saw last night at the dinner, who ran the CDKL5 and Rett clinic at Boston Children's, and he's now head of all rare diseases at Roche. They are also interested now and discussing with us about potential fundamental genetic approaches to increase CDKL5. Ultragenics, a fantastic rare disease-focused company led by Dr. Emil Kakis, again a wonderful human being. They recently now have an interest in the disorder. Zogenix, fantastic results with Dravet syndrome, with fenfluramine, who are also now interested in this disorder. AMO Pharma is a company, it's a British company, and they actually are interested in the tianeptine, which you heard about, that antidepressant that might be repurposed for the disorder, and Sage Therapeutics, that again has a series of drugs, neurosteroids, that are potentially interested as well. And there are some that are not on the list. So none of those were interested or act. So all of them are either currently active or interested to get active in this disorder, and that was not the case for any of them 3 years ago, 2.5, 3 years ago. So the field has moved a long way, and we're certainly optimistic. Engaging enabling industry, sorry for taking so long with that, but I think it's important. Breaking down the barriers, so between industry and academia and the patient community. And plugging the gaps. So I'm not going to go into the details of these good slides, mainly because I don't fully understand them. If you have questions, that's what Dan and Anna, I'm sure, would be happy to help. But basically, it's a combination of funding the basic science to understand the disorder, but in parallel, funding the toolkit, building the toolkit that's going to help encourage more researchers to get engaged and companies to take an interest. Thanks to IFCR, there were already mouse models and cell lines, but we've tried to expand that as well. We're partnering with IFCR on a new iPS cell line project, as well as to expand the network of clinical centers of excellence that are so important as sites that are ready for clinical trials, and to help build up the natural history of the disease. Multiple therapeutic approaches. So we're really interested in going across the spectrum, betting on every horse, and hoping that several horses are going to cross the finish line. And we don't know in what order, which is why we don't say cure, we say cures. In 2015, at our first CDKL5 forum, which is the Science Symposium, we set the goal of treatments within 5 years and cures within 10. So that's treatments or therapeutics that are going to help with the symptomology, such as hopefully ganaxolone and others by 2020, but cures, i.e. disease-modifying treatments that will address the broader spectrum of symptoms and probably have to target CDKL5 itself, either at the genetic level or the protein level, by 2025. And we think that's achievable, but we can't do it alone. It has been a team effort and will continue to be a team effort. I'm not going to go into detail, but you've heard about gene replacement therapy, enzyme replacement therapy, evolution of gene editing, drug screening approaches, drug repurposing, bioinformatics, so really or X-ray activation, the whole spectrum, and sometimes combinations. The community building and stakeholder engagement is absolutely key. A lot of these things you've heard about. So the CDKL5 Forum is an annual meeting of scientists, researchers from academia as well as industry. We had the third meeting in November in Boston, and we had almost 200 attendees, including a third of the audience were from industry, from 35 companies. So about 3 times the list you see there. And we also have an online portal for the researchers, but almost 500 members now from around the world. They can talk to each other, explain protocols, tools, reagents, answer questions, and so on. So really trying to create, science is so important, but it tends to happen in individual silos. So we're trying to link people together as much as we can. Just quickly going through the key milestones of the journey so far. We started in mid to late 2015, just over a year after Alia was born. We're now funding or have funded a total of 31 separate research projects in 41 labs at 30 leading institutions, so over 120 scientists in total. And the key is also to enable bigger funding. So by funding some pilot projects, some early grants, you can hopefully unlock even more funding. And we're one more update actually that Karen reminded me to announce. Thanks to some early research that we co-funded with IFCR and George Zhao's lab at the University of Pennsylvania, he was able to get very recently the first R01 research grant from the NIH. So that's about actually $2.5 million over 5 years. And that's thanks to all of your tax dollars, so thank you for that. So that's the first major grant. There was one other that included CDKL5, but this is just CDKL5. We hope for many more. NIH, NINDS, which is the Neuro Institute at NIH, they recognize they need to do more in this space. And it's not because they had bad intentions. In the past, as we all know, it was considered part of Rett syndrome. Now that's known, it's a distinct clinical disorder. So they want to do more in that space. So we're very hopeful. So engagement with the FDA and the EMA, that's started thanks to Marinus. We hope to get more organized, and with IFCR have a formal meeting with the FDA to better educate them about the disorder and work to defining endpoints with the clinical centers and the patient community. So it starts and ends with the patient. This is Alia, slightly more recent photos, but she's our world, but she's one of tens of thousands of our kids who are suffering with this disorder around the world. And we're really doing this for all of them. And we can't do it alone. It needs the partnership with industry, as we mentioned, with the clinicians, with the academic research, and with the patient community. And really, we need all your help. First thing to do is what you're doing. Love your kids and each other. I think that's the most powerful thing we can all do. and support your national patient associations. Here in the US, IFCR and the other national ones who are present here, because we need the work that they're doing. It's critical in terms of patient identification. You can raise awareness of your disorder and help your pediatric neurologists to understand that there's a lot going on. They need to be diagnosing these kids earlier and informing the different national associations. Because the bigger the community we have, the more clinical trials we have, the more industry interest we will get, and we know that they're out there. They just need to be identified and included. So I'll stop there. I just want to quickly mention also CDKL5 UK. Caroline, where's Caroline? I know she's here. It's been great working with her in the UK and raising the awareness there, and we've funded various projects there. Antonino Caridi and his family and the amazing conference they put on. Where's Antonino Massimiliano? And I mentioned Mike Jasulavec, who really has been a key advisor, and a lot of, PTC, Amicus, so much of that corporate interest is thanks to the work that he did. So I think it was 20 minutes. So we have hopefully 10 minutes for questions, if there are any. Anything at all, happy to take them.
Questions for the Lulu Foundation, questions for Tim, questions about research, development, global community.
Anything you guys want to add?
No? Yes.
Anything you guys want to add, or did I miss?
So I think you really nicely pointed out one of the big problems, that we're not identifying many of these patients. And so much of that is dependent on things out of our control, insurance, standards, and how we get genetic testing paid for, wide range of clinicians that are doing certain things. And in many ways, those of us who think about precision medicine a lot, this is a fundamental issue that is a huge barrier. And I'm just curious if we're thinking about ways that we can start to try to address that. And because it's such a big challenge. You can't treat something you don't know that you have.
So I don't think there's a magic bullet, but I think the trend, because the cost of sequencing has come down so much, and insurance companies are covering it here, or national health providers in Europe, and there's more awareness of the disorder. But sometimes, neurologists or pediatricians just think that, treating the seizures might be important, but maybe diagnosis isn't immediately important. So I think it's on us to raise the awareness with the clinicians, and the fact that there is a patient community, there are clinical trials coming, there's a lot of research going on, and there is really hope, and we really do believe that. And I think but not just to diagnose, but then once they diagnose, to actually refer them to the patient organization. I mean, most will find them anyway, but some may be reluctant. I mean, early on when you have the shock and the sadness, you don't want to talk to anybody. But it's important to understand, just get on their list, even if you don't want to engage just yet, because there's a reason to. There are trials coming, and there are advances that are being made. Yes, and there are other programs now to accelerate, like Invite, who are here as well. They've partnered with industry, and they're sequencing kids with epilepsy from the age of 2 to 4. Now we want the 0 to 2 to be covered as well. That was for a particular clinical program, and so now we've encouraged them to do that, and they're starting to find the corporate sponsors who are interested to fund that part as well. So that will accelerate further the diagnosis rate. Is that all right? Good. Well, thank you very much. Great to be here.
Thank you. Okay, so just some quick housekeeping. If you are in your app or if you're on the program, we've got the 9 a.m. breakout sessions. So we've got movement disorders and CDKL5 syndrome in Aurora 6, okay, which is Dr. Suter from Texas Children's Hospital. And then in this room, we've got a very good conversation about building your care team with Tristan from the Colorado Children's Hospital, talking about how we need to have a multidisciplinary approach to CDKL5 treatment. In addition, we also have the children's activity room on, and then we'll regroup here at, let's see, so we'll have morning refreshments at 10 and then regroup here at 10:15 for multiple other sessions in this room and then also in Aurora 6. Have a good morning.